Right. Hi everyone. So I'm going to talk to you about comparative taphonomy of embryo-like fossils from the Ediacaran and Cambrian of South China. So molecular clocks estimate that the origins of the Metazoa fall somewhere within the Neoproterozoic, but the first definitive animal fossils that we see don't occur really until the Cambrian. And so the Ediacaran Doushantuo biota from the Doushantuo Formation of South China is therefore of great significance because it is a window of exceptional preservation within this sort of time frame that molecular clocks estimate animals to have appeared. So the biota preserves a variety of forms, of which that is dominated by these millimeter scale embryo-like fossils of the Tianzhushania complex. So we have sort of one cell megasphaera, and then 2, 4, up until 32 cell Parapandarina, and then Megaclonophycus. And these are of highly debated affinity. They're currently considered to be multicellular metazoans, but they've also been described as non-metazoan holozoans and as chlorophytic algae. And so one way we can try and test what their affinities might be is to compare them with fossils that we know to definitively be animal embryos. And one deposit that provides us with such fossils is the Kuanchuanpu Formation of South China. So the Doushantuo and the Kuanchuanpu are in the same succession. So you have Doushantuo here, Ediacaran, overlain by the Donghue, and then the Kuanchuanpu lies on top of the Donghue. So they're very similar deposits. They both consist primarily of limestones and phosphatic limestones. And the fossils in both are preserved in calcium phosphate. So they have they've undergone very similar diagenetic histories. The Kuanchuanpu fossils themselves, again, were a variety of different bits and pieces, of which the most important ones for this, obviously, are the embryos. And we also find the adult stages of what these embryos grew into. And we see pretty much a whole ontogenetic sequence preserved, so we can be pretty certain that these were indeed animals. So these are the Kuanchuanpu embryos. There's 3 broad types. We have the olivoides here, pseudoides, and then the so-called cleavage embryos, which are associated with both of those previously mentioned forms. So in terms of the comparison, so we actually see them preserved, Doushantuo and Kuanchuanpu, at different stages of decay. In the Doushantuo, we almost always see distinct cells preserved. And in some cases, such as in this Parapandarina, we see the intracellular structures preserved. In the case of Parapandarina, these are interpreted to be lipid granules. In Kuanchuanpu, by contrast, we never get any intracellular structures preserved. What we have instead is that we can see that the fossils of the organism had started to coalesce, and we see the beginnings of the breakdowns of the boundaries between cells. So both sets of fossils are primarily preserved in 2 different mineral phases. So you can see there's a dark mineral phase, which is an early forming phase that replicates original biological structure, which in the Doushantuo preserves the cell contents. So in this Megaclonophycus, it's actually preserving the component cells there. And then in Parapandarina, it's primarily these lipid granules in this cell, and more cytoplasm-like stuff in that one. And in Kuanchuanpu, we see that the dark phase replicates the cell membranes, but not the cell contents. And then the second phase is this bright phase, which is later void fill, which in the Doushantuo fills in around the edges. So if we get membrane preservation in the Doushantuo, it's always with this later void fill. Whereas with the cleavage embryos, when we see the cell contents mineralized, it's always, again, the void fill, and not the phase that replicates biological structure. And so we've also done some EPMA analysis on this. to try and sort of confirm that we are seeing the same phases, and seems pretty clear, at least to us, that we've got the dark phase, which is you can see in this cleavage embryo on the nice this sulfur map is picked up quite nicely here, and then again sort of in some of the in the cell contents sort of in the megaclonophycus. So the dark phase is enriched in sulfur compared to the bright phase, and also somewhat enriched in carbon, although there is very little carbon generally in these. But the dark phase is then depleted somewhat in both calcium, phosphorus, and fluorine compared to the bright phase. So we're clearly seeing the same two mineral phases at both sites, but they preserve very, very different things in each set of fossils, which is interesting given that if they are fossils of similar sorts of things, you might expect to see them preserving the same sort of stuff, but they don't, and it's always preserving different things, membranes in Kuanchuanpu and cell contents in Doushantuo. So we have sort of a summary comparison of the decay pathways. So in all cases in Doushantuo, the initial mineralization is of the cell contents, sometimes with interior structures preserved, sometimes not, and then it's later void fill that fills in the membranes. And then with Kuanchuanpu, the membrane tends to persist, and we get coalescence. We get early mineralization of the membranes, and then later void fill of the interior. And what we do also sometimes see with this one, which again we don't see with Doushantuo, is that sometimes the membranes don't actually mineralize at all, and it's just the contents, and then falls apart. And so what we sort of see and what we think this is telling us is that we have deposition of mineral phases that show a consistent pattern at both sites, but the Kuanchuanpu embryos are preserved in a very different way to the Doushantuo embryos, with structures present in one that are entirely absent in the other. And we think this could reflect differences in the biology of the organisms, and we think perhaps that if we have this otherwise very similar preservation that is preserving in what we know to be definitively one thing in what we know to be definitively animal embryos, but entirely different things in these other ediacaran embryos, then that might suggest that they are not metazoans as people believe, but something else entirely.